Thank you very much. It's my great honor and privilege to be given the opportunity to speak at this important event. I'd like to share with you some of my journey as a scientist who has done a little bit of entrepreneurship. Can I have the next slide, please? So this is a picture of myself actually taken over 30 years ago when I was still a medical student in Oxford. Thank you. Next slide, please. So as a medical student, we have to study different subjects. And one of those subjects is obstetrics. So I learn about how when a woman is pregnant, she would like to know about the health of the baby. But at that time, if we want to test the DNA or chromosome of a baby, we have to stick a big needle into uterus in a process called amniocentesis. But every time we do this, there's a risk we might harm the baby. So as a result, many expectant mothers become very worried. Okay, next slide, please. So as a young medical student, I was thinking, is it possible I can get rid of all of that by developing a new blood test, which I can do from the arm of the mother? Next slide, please. But of course, the problem is that thinking was against conventional belief because we all know that our blood and our mother's blood are separate. For example, we could have different blood group types as our mother, but I was thinking maybe that conventional belief is not 100% accurate. So maybe there is a chance that the baby might release its cells in the bloodstream of the mother. Next slide, please. But the question is, how do I know that this cell is from the baby? So after thinking about it for a while, I thought that if the baby is a boy, maybe I would have a chance to actually see the male fetal cells floating in the blood of the mother. Next slide, please. And then next slide, please. So I decided to actually, again, to try it out. So I actually called a number of professors in Oxford and asked them, I have this idea. But of course, you can imagine that most of them would laugh at me because they thought if this phenomenon exists, people would have found it before. But anyway, I decided to press on. And eventually, we did an experiment like this. So I managed to find a dye or probe that will stick to a male cell, and then you can have a black dot. So you can see in this blood film of from a pregnant mother, there's a number of male cells, so showing that my thinking is correct. But then I thought, well, if every time you have to do a prenatal test, you have to go to a microscope to hunt for those cells, then it's not very efficient. So I want something faster. Next slide, please. And then one day, I went to a lecture by this gentleman who is Professor Sir John Bell, who is the Regius Professor of Medicine in Oxford. So he was talking about a new method called PCR. Next slide, please. So basically, PCR is a method which will amplify DNA from one molecule to millions of molecules. So during the COVID pandemic, when we do the nucleic acid test, that is basically a PCR test. So he said that this method is going to change the world, and I believe him. So after the lecture, I went to him and asked him to teach me how to do this method, which he graciously did. Next slide, please. And so I managed to actually show that by using PCR, I can very simply see the male chromosome from the baby boy. Next slide, please. And then we published this in this famous journal called The Lancet. Next slide, please. And a few months later, The Lancet actually published an editorial called Is It a Boy? Because you can actually use this method theoretically to tell the sex of a baby. Next slide, please. And then I graduated and then became a junior doctor. So in those days, as a junior doctor in the UK, you have to work maybe 120 hours a week. But in my spare time, I still think about this project. I thought that because I've started it, I have some duty to actually finish it. So I decided that after this 1 year as a husband, then maybe I should go back to Oxford to do this full time. So next slide, please. So you see this picture is a picture of myself as a PhD student in Oxford. You can see that I'm sort of all completely wrapped up, almost like a terrorist. And the reason is because I'm actually using the male chromosome as a marker. But every cell in my body has a male chromosome. So basically, I'm protecting my sample from myself. Next slide, please. But sometimes in life, when you do this part-time, it's all fun. But if you do it professionally and full-time, suddenly all the problems emerge. And this is one of those things. So when I start to work on this project full-time, suddenly I find that even though the fetal cells are there, but the concentration is super low. It's almost like trying to find a needle in a haystack. So next slide, please. So eventually my scholarship ran out, so I have to face reality and to finish. So at that time, the science didn't go well, but extracurricularly, I managed to meet my wife. Next slide, please. So both of us actually graduated together in Oxford in the summer of 1994. And next slide, please. And then I went back out to be a doctor for a few years. And then 1997 came, and then we thought, well, it's time to come back to our home. Next slide, please. And so I joined the Chinese University of Hong Kong. Next slide, please. And then when I resigned from Oxford, there was a period of like 3 months. I was thinking, well, what have I done wrong? Because previously I've spent 8 years trying to look for those elusive fetal cells, but couldn't get to work. So what happens? And then about 3 months before I came back, there was in this journal, there were 2 papers, which says that in cancer patients, you can find cancer DNA in the plasma. Next slide, please. So what is plasma? So basically, when you take blood from us, the whole tube is red. But if you put it on a table for a couple of hours, the red blood cells will go to the bottom, and then on the top there is this fluid, which is yellow in color, called plasma. So I was thinking, maybe the previous 8 years, I was looking in the wrong place. Next slide, please. So instead of looking at a cell, maybe I should be looking in this fluid. Next slide, please. But this thinking was very against conventional belief because you can imagine the DNA is the brain of a cell. And instead of finding the brain inside a cell, I was trying to look for it outside. It's almost like for those of you in the banking industry, you are instead of finding the money in your safe, you actually just have a money lying outside the safe. Next slide, please. But anyway, so I decided that maybe since I have come back to Hong Kong after start from scratch, that's a time to try something risky. But in those days, in 1997 in Hong Kong, the funding for research is nothing like now. So basically funding, we are talking about maybe $500,000 per grant. And I have not a lot of resources to do experiment. And I never handle plasma. So how do I extract the DNA out? Next slide, please. And then suddenly, next please. And then suddenly I recall that in my years in the UK, I never actually enjoyed UK food, right? So I'm quite a Chinese preference in what I eat. So sometimes when I had enough of college food, next slide please, I will actually go back to my room to cook instant noodles. And for those of you who share my interest in instant noodles, so what you do is that you boil some water and you put your noodles in. And a few minutes later, you have your meal. And I was thinking, how about if I take some plasma and just boil it for a few minutes and take a few drops of that soup to be tested? You probably think you're crazy, right? But if you don't have money, this is what you have to do. Next slide, please. And surprisingly, even with this very crude preparation, I was able to get a Y chromosome signal in some of the samples. Like you can see from the left-hand side, there are 3 women with 3 signals, and then there are 4 women with no signals. And eventually, when I look at the sex of a baby on delivery, I was surprised. Basically, all the women with signals all have baby boys, all the ones with no signal have baby girls. So this is the first discovery of fetal DNA in maternal plasma. Next slide, please. And then we find that this fetal DNA is present very early, from maybe 6 weeks of pregnancy onwards. Next slide, please. And by the 10 weeks of pregnancy, the concentration reaches 15%, which is amazing because imagine 10 weeks, it's like 2.5 weeks. The baby is very small, but the mother, of course, is huge in comparison. And yet, the baby is working overtime to push all this DNA into the blood of the mother. Next slide, please. So I was thinking, maybe I can actually try to develop this into a medical test. Now, the number one reason why pregnant women want to go for prenatal testing is because they're worried that the baby might have chromosome disorders like Down syndrome. So basically, in a healthy cell, we have 23 pairs of chromosomes. And their number from 1, 2, 3, 4, 5, 6, etc. And number 1 is the longest chromosome. 2 is a bit shorter. So chromosome 21 is a very small chromosome in which, in a Down syndrome baby, instead of having 2, you have 3 of those chromosomes. Next slide, please. So normally, when you try to diagnose Down syndrome, you have to take a cell from a baby, and then you probe it, and then you count how many chromosome 21 it has inside the cell. So in this case, you can see 3. So this baby has Down syndrome. Next slide, please. But in my field, we have a problem because I was looking at DNA outside cells. So how can I look at something outside cell to deduce what is inside? It's almost like we're trying to look at how many people is outside this ballroom to deduce how many people is inside a ballroom. It's quite challenging. Next slide, please. So I've been thinking about this for about 10 years. But luckily, during that time, a Chinese university didn't fire me. So eventually, after 10 years of thinking, I finally arrived at the answer, which seems very simple. So imagine you have a wallet, and sometimes you have 4 coins in a wallet. Next slide, please. Some other times, you only have 3 coins. But I want to know how many coins you have in your wallet today. And of course, the simplest way is to ask you, hand me your wallet. But that's a little bit intrusive, like invasive testing. Next slide, please. But another way to do it is to ask you to stand on a balance. Now, theoretically, on a day when you're a little bit heavier, when you have more money, you should be a bit heavier, provided your balance can be very accurate. Instead of, say, 70 kilograms, I can measure 70.01 kilograms. So the question now is, how do I develop a molecular balance for DNA? Next slide, please. So I was thinking, well, it's difficult because normally when you take a sample from the mother, most of the DNA inside is the mother's, which is a red dot. And occasionally, you see a fetal DNA, which is the one in yellow. So when you're trying to test it or count it, the mother signal is going to overwhelm the baby signal. Next slide, please. So I was thinking I can overcome that if I don't do the testing together in one go. I try to distribute all this DNA into multiple tubes so that one tube will only have one molecule or no molecule. This is kind of like digital, like your computer, 1010. Next slide, please. So under this situation, the baby's DNA can be analyzed on its own without interference from the mother. Next slide, please. Next slide, please. So we managed actually to do this. So we cracked this in 2007 and then published this paper. So I was very happy. But strangely, next slide, please. A few weeks after we published this paper, I saw another group publish something very similar to us. Next slide, please. And also very strangely, the 2 papers were submitted on the same day, which is incredible, right? It's almost like when you think of something, somebody else think of something. Next slide, please. But of course, that means that we have competition. But on the other hand, it also is good because it means that our thinking is probably correct because 2 groups have independently arrived in the same result. Next slide, please. So we thought we're going to crack this test by using this approach, provided we are fast. But then we run into some obstacle. So what's the obstacle? The obstacle is that if I'm trying to do a PCR test, you will record a COVID. Then like for COVID virus, we have to find a gene in the COVID, like the S protein, the spike protein, and then do a PCR on it. But the problem is that chromosome 21 has 150 genes. So you just focus on one gene, you are forgetting the data from 149. So it's not a very good test. So we want something more efficient. Next slide, please. But at that time, a new technology just came in, which was marketed by a company called Illumina, which is basically doing a type of sequencing which is 1 million times faster than before. Next slide, please. And normally, you can imagine if you were in my situation, of course, we would try to get this technology ASAP. But unfortunately, at that time, in a university system, we have some problem. Next slide, please. Because at that time, the University Grants Committee have an interesting rule. They says that if you want to buy something expensive, all the other universities have to agree. It's just imagine you are doing some big business deal and all your competitors have to agree before you can do it. So you'll never have that deal. But of course, I'm pleased to say that now under Dr. Tim Lui, this rule is no longer here. So next slide, please. So at that time, I've been waiting for 10 months and no progress. So eventually, I decided I need to do something else. So I called up this guy who is the CEO of a NASDAQ-listed company called Sequenom. And I said, well, we have this project. Can you help us? So within 5 minutes, he said, no problem. Here is your machine. So next slide, please. But of course, in this sort of deal, there's always a cost, right? Because you have to license the technology to them. So then we eventually actually developed this test in which I randomly sequence the DNA in blood, map it to the chromosome, and then calculate whether each chromosome is normal or if one chromosome is higher. Next slide, please. When I did this, I find it's very good. You can see the green signal and red signal are from Down syndrome babies. And the signal is very high compared with the blue signal and the orange signal, which is from normal baby. So we did 28 cases, 14 with Down syndrome, 14 normal. The test is 100%. Next slide, please. So then we published the work in the year later, 2008. And of course, once again, very happy. Next slide, please. But unfortunately, within a few weeks of that, the other people published the paper as well. And then I was thinking, wow, who is this guy? He's keep chasing my tail. So then I take a look at his website. Next slide, please. And I find that he's a gentleman called Steve Quick, who is actually from Stanford. Next slide, please. And very strangely, he also actually studied in Oxford and graduated in the same year, 94. And so I went home and spoke with my wife. I said, well, is this person, what do you think? Next slide, please. And let's dig out this photo. Next slide, please. And that's me and my wife. Next slide, please. And that's Steve Quick. So it's almost like tonight when you listen to me, and the person sitting next to you is the person who's going to compete with your next deal 5 years from now. But of course, in this room, it's probably more likely than at that time, right? So next slide, please. And interesting, the story didn't end there, because a few years later, I was fortunate enough to be elected into the US National Academy of Science. So on the day when the result came out, I saw my name, Low, so very happy. And then scanned it down, I saw his name. The next slide, please. So 3 of us decided to actually take a picture. But you can see from this picture that the environment in Stanford is probably more stressful than at CHK, right? So next slide, please. So anyway, so now we seem to have this technology which seems to work, but then we need to basically push it out to the clinic. But before I do that, I need to do a big clinical trial. So we spend the next 3 years doing that. Next slide, please. So eventually, after the clinical trial is done, we find that the test is 99.7% accurate. So we think we're ready. Next slide, please. But of course, if you want to ready to do technology transfer, the first question the company will ask you is, do you have any patents? Next slide, please. So because of this reason, for the last 20-odd years, we've been very diligently filing intellectual properties. Next slide, please. Next slide, please. And every year, this magazine called Nature Biotechnology will rank the top 20 groups in the world with the most biotechnology patent. And I'm pleased to report that for the last few years, I've been on the list. Next slide, please. For example, in the latest list, I was number 7 in the list. Next slide, please. And of course, you wonder, who are the other guys on the list? Next slide, please. So number 1 is a gentleman called Jiang Feng from MIT. He was actually here one month ago. He was one of the inventors of a new technology called genome editing, CRISPR. Next slide, please. And number 2 is the CEO of BioNTech, of course, mRNA vaccine. Number 3 is Bob Langer, who has started many companies, including Moderna. Next slide, please. And number 5 is Jennifer Doudna, who is also inventor of CRISPR and who got a Nobel Prize in 2020. Next slide, please. Next slide, please. And then another gentleman from my lab is number 13. Next slide, please. So when you go through a list, you find actually that most of the top inventors in the world are still from the US. And here they list 3 from China and 1 from Europe. So I think if we want to make further contribution to the technology field, we need to work harder. Next slide, please. But that is now. But at that time when I invented the test, I wasn't as savvy in terms of intellectual property. So at that time, our first reaction is to license the technology out to some company. So at that time, our university licensed it to a UK-based company, which didn't realize the significance of my work. So they sat on it, and then after a few years, they thought it was rubbish and gave it back to us, free of charge. And of course, for those of you who know this field, this is a big mistake. And of course, a few years ago, I met that manager who is no longer with that company. So anyway, so next slide, please. Next slide. So I have to find another company which is interested. And this is when, next slide, please, when I came into this company, Sequenom, that I mentioned to you just now. So the gentleman standing next to me is Charles Cantor, who is the Chief Scientific Officer of this company, Sequenom. So we license our technology to them. Next slide, please. And you can imagine, of course, in Stanford, the professors are probably more technology savvy, and so there are many startups. So Steve Quick actually licensed a company to a company called Veronata. Next slide, please. And then Veronata got acquired by Illumina for about 600 million US. Next slide, please. So now you have Sequenom with my technology and Illumina with their technology. And so usually company, whenever they have technology, they use it like weapons to fight. So there's a series of 4 lawsuits, and eventually I won 3 of them, and they won 1. Next slide, please. So then we decided to actually settle out of court. Next slide, please. So eventually, basically what they did is that they pay us 50 million US, and then we pool all the patents together. So anybody who want to do a test, just license from this pool, and there we go. Next slide, please. And next slide, please. So now this technology is called NIPT, non-invasive prenatal testing, has been done over 100 million times. Next slide, please. So up to now, you can see why I come into this technology is because I was inspired by the cancer field. But then I thought, well, the prenatal field seems to be going very well, so maybe it's time to use what we've learned into cancer. Next slide, please. But of course, for cancer, I cannot use the simple technology yet, like using a Y chromosome, because it's all within one person's body. Next slide, please. So then we decided to use something called epigenetics. Now, genetics is about the coding on the DNA, A, C, T, G. But epigenetics is if the coding doesn't change, but you have some sort of modifications which changes, almost like the decoration in this room. Next slide, please. Or is like the formatting of your Word document. So your font can be different font size, different color, underline, bold, etc. So as it turned out, the DNA can be modified or formatted in different ways. Next slide, please. And cancer patients have a special type of formatting. So eventually, we developed a test in which one test can be used to detect the formatting of multiple types of cancer. Next slide, please. And of course, after you tell somebody that he or she has cancer, the next question they'll ask you is, where is the cancer? Next slide, please. So as it turned out, the different organs in our body, the DNA is formatted differently. So I can also use the same technology to tell us where is that cancer. Next slide, please. And this technology was actually named by MIT Technology Review to be one of the top 10 inventions of 2015. Next slide, please. And this time, I'm kind of a little bit more commercially savvy. So instead of licensing out, I decided to start my own company called Serena, which stands for Circulating Nucleic Acid. So basically, our mission is try to find cancer early, and so that we can take some action. Next slide, please. But interestingly, in the Silicon Valley, Illumina just started a company called Grail. And they started it with 100 million US, and also with the same sort of mission to detect cancer early when it can be cured. But interestingly, at that time for Serena, I have the technology, but no money. But for Grail, at that time, they have the money, but no technology. So then eventually we decided to talk. Next slide, please. Next slide, please. And then in 2017, we merged together, and I became the scientific co-founder of Grail, and then our technology became Grail's technology. Next slide, please. And so this is a picture taken at the ceremony to celebrate our merger. The background is the NBA final of 2017. As you may recall, in that year, actually, the Golden State Warriors won. But when we booked this room, we have no idea they're going to win. But anyway, book it. And then the guy standing next to me is a guy called Jeff Huber. Some of you may know him. He actually used to be at Google. He's the person who brings us Google Maps. But unfortunately, because his wife passed away of cancer, then he decided to use his expertise into cancer. Next slide, please. And after a few years of work, Grail basically used our IP to develop this test called Gallery test. So basically, one test can detect 50 types of cancer. Next slide, please. And this test was chosen by Time Magazine to be one of the top inventions in 2022. Next slide, please. So it's called Cancer Finding Super Test by this magazine. Next slide, please. And of course, you would say, well, so how good is this test? Now, so for example, in this publication from Grail, you can see they looked at over 6,000 people with the median age of 62 years. And basically, if the Grail test is positive, the chance of that person having cancer at that minute is 75%. If the Grail test is negative, the chance of that person not having cancer at that minute is 98%. And the accuracy of us telling you where is the cancer is 85%. You can see those, so this parameter looks very good. But still, we have some weakness. Next slide, please. The weakness is that if it's stage 1 cancer, we can only detect 24%. Stage 2 is about 56%, and then rising from stage 3 and stage 4. But of course, if I'm unfortunate enough to have cancer, I'd rather be picked up early than late, at least have a chance. Next slide, please. So we still have to make the early detection better. And also, currently, the test is still quite expensive. It's about $950 in the US. But I think for example, mainland China and some other parts of the world, we want something cheaper. So I've recently started a new company called Insighter with a technology which is several-fold cheaper. Next slide, please. So anyway, in summary, so I've chosen a path to become a clinician scientist. So my kind of a bridge between medicine and science. Because if I'm just a clinician, then no matter how hard I work, every day I might only serve 50 to 100 patients. But as a scientist, if I develop something that works, maybe I can actually help millions of people every year. And secondly, being a scientist, I think that we have to somehow merge science with daily life. Because every day, there might be inspirations. Now, for example, like I was inspired about the original test from cooking noodles. And maybe even tonight, speaking with some of you, I might have some other inspiration. And also another thing is that breakthrough might come if originally you think that something is not related, like pregnancy and cancer. But if you can see the link between them, then suddenly the new field is started. And also, it's also important for us professors not just to work in university, but to think of a way to translate this down commercially. Because if I'm just in the university side, my test will only reside in the library. But now it can actually benefit patients. And finally, I think the policy is very important. For example, just now I talked about the policy about asking permission from all the other universities, even though that took me 10 months, but it actually cost me several years of litigation. And some other policy would be like, for example, like Hong Kong, we are only a city of 7.5 million people. If I want to do large-scale clinical trial, I have to rely on maybe the Greater Bay Area. But if that's the case, we have to look at the easy transfer of samples and data across the border. And there are many other similar policy issues we have to think about. Next slide, please. And finally, actually, research is not just myself. I have a whole team that every day we brainstorm, we encounter hardship and success together. Next slide, please. And thank you very much for your time.